Hello students, this module is for the students of Bachelor of Science first year. Subject is Botany, Semester 2. Course code is BOC 102, course title Biodiversity 2. The title of the unit is Pteridophytes. Name of the module is Sexual Reproduction of Pteris. Myself, Ms. Prachi Verenekar, Assistant Professor, Government College of Arts, Science and Commerce, Sanklim, Goa. In this module, we will be dealing with sexual reproduction of Pteris. At the end of the module, you will be able to explain the sexual reproduction of Pteris. You will also be able to describe the structures of antheridium, spermatozoid and archegonium in Pteris. You will also be able to explain the fertilization in Pteris. Let us start. The adult prothallus is small, green and photosynthetic. It is dorsoventrally flattened, aerial, cordate type and heart-shaped. Its size varies from 6 to 13 mm in diameter. The cushion-like deep apical notch is present at anterior end. The growing point lies below the apical notch. As you can see in the diagram, the growing points are present towards the upper portion. The apical notch is made up of meristematic cells. The prothallus is autotrophic. That means it is having the ability to prepare its own food and it is independent in nature. The prothallus is made up of polygonal or hexagonal thin-walled parenchymatous cells, as you can see in the diagram, which is one cell thick towards the margin and many cell thick towards the central cushion. Many delicate brown hair-like unicellular rhizoids are present on the lower surface of the posterior side of the prothallus. These rhizoids help in attachment as well as absorption of water and minerals from the soil. The prothallus is autotrophic, the haploid plant of the gametophytic generation. Moving on to the next, that is the sex organs. The prothallus is monoecious and bears both antheridia and archegonia on the ventral side. The antheridia, that is the male sex organ, are found between the rhizoids towards the posterior side of the prothallus. The archegonia, that is the female sex organs, are developed on the central cushion near the growing point. Next is the structure of antheridium. The antheridium is small, sessile and globular structure. The antheridial wall is made up of three cells. The first ring cell, the second ring cell and the opercular or the cap cell. As you can see in the diagram, there are numerous spermatozoids present in the center which are large, coiled and multiciliate. The cap cell forms a lead, the second ring cell forms the middle portion and the first ring cell forms the basal part of the antheridial wall. Before moving to the structure of spermatozoid, we need to know how the spermatozoids are actually liberated from the antheridium. As you can see in the diagram of the antheridium, once the antheridium is matured completely, it starts absorbing a large amount of water. When the antheridium absorbs a large amount of water, the cap cell breaks open and it releases out the spermatozoids which are present inside. This is how the liberation of the spermatozoids takes place. Moving on to the structure of spermatozoid. Each spermatozoid is large, multiflagellate coiled structure. It is made up of motor apparatus and the nucleus. The motor apparatus consists of numerous basal granules or centrioles and a mitochondrial part, and the flagella arises from this part. A cytoplasmic sheath surrounds the coiled nuclear portion. The vesicle portion is composed of homogeneous ground material of flat globules with 4 to 6 plastids and small granules. Moving on to the structure of archegonium. Each archegonium is flask-shaped and consists of swollen basal venter and a narrow curved projecting neck. As you can see in the diagram, it is a flask-shaped structure bearing a venter which is broader in nature and a neck which is very much narrower. The neck is composed of 4 vertical rows of cells and each row is 3 to 7 cells in height. The neck contains a binucleate neck canal cell pointed towards the antheridium. The venter contains a single large egg and venter canal cell. The venter wall is absent because the venter is embedded in the tissue of the thallus. The next is fertilization. Fertilization takes place with the help of water, which occurs between the lower surface of the prothallus and soil. The disintegration of venter and neck canal cell at maturity of archegonium results in the formation of mucilaginous substances. Mucilaginous substances contains malic acid which attracts the spermatozoids. The spermatozoids swimming actively in a layer of water are attracted towards the open mouth of the archegonium. Many spermatozoids reach the open mouth of the archegonium. The number of spermatozoids, which are also called as antherozoids, may enter the archegonium, but only one fuses with the egg. As you can see in the first diagram, the spermatozoids are entering from the neck of the archegonium and they are reaching towards the egg cell in the ventrum. The haploid nucleus of the spermatozoid fuses with the haploid nucleus of the egg, forming a diploid zygote, which can be seen in the second diagram. Once the fertilization takes place, the zygote further divides to form the young sporophyte, as you can see in the diagram. These are the references which I have used for the module. Thank you.